Very good morning, dear students. We have started unit number 4 of biochemistry, and this unit was the continuation of the previous unit where we have discussed the important aspects of proteins. And one important types of protein, as you all know, that is known as enzymes. All enzymes are protein. The previous class we have discussed the general characteristic and properties of enzyme. Then the next important aspects we should know before the functional aspects of enzymes are the classification and naming of the enzyme. The classification of enzyme. So enzymes are one of the most important types of biological catalyst which are completely different from the chemical catalyst and which speed up the biological reactions. Okay, and in an introduction, this enzyme need to be named, and there should be so many criteria for the classification of enzymes. So the major criteria are used to classify and name the enzymes are as follows. The first one is the substrate acted upon by the enzyme. So the substrate acted upon by the enzyme, the name of the nature of the substrate in which the enzyme is acting. Next one is the type of reaction they catalyzed. Okay, the types of biochemical reaction they catalyzed. Third one is the substrate acted upon and the type of reaction they catalyzed. So both of the first and second one is the one comes together. The substrate acted upon and the type of reaction they catalyzed also involved into the classification. The next one is the substance that is synthesized due to the enzymatic process. What is the substance or the end product that is being synthesized? Okay. Then the next one is the chemical composition of the enzymes. Okay, so we are knowing that the enzymes are protein, and what are the major chemical composition that is present in the enzyme. The next one is the substance hydrolyzed and the group involved. Okay, so what is the substances they hydrolyze and what are the major groups involved in this particular process. And these are the fundamental aspects. The major classification criteria is proposed by the International Union of Biochemistry, or IUB. They made a detailed classification based on all these peculiar features, the overall chemical reaction taken into consideration. So these are an overview of the classification criteria of enzymes. So now we will discuss one by one in detail. The first one is the substrate acted upon by the enzymes. Okay, so any substance this acted upon which an enzyme is known as the substrate. Okay, the substance upon which an enzyme acting on is known as substrate. Okay, and in 1883 a scientist called Duclos. Okay, so Duclos named the enzymes by adding there is a suffix ASE, the name of substrate catalyzed. So which substrate the enzyme acted upon, and along with the substrate simply add the ASE as a suffix to denote the enzymes. Okay, so say for example enzyme acting upon carbohydrates. Here carbohydrates are the substrate, and the enzyme of course carbohydrates. Okay, you can see carbohydrate, they have used the ASE, carbohydrases. Then the enzyme acting upon protein, so proteinases, so protein and the ASE proteinases. Then the enzyme acting upon lipids are lipases. Then the enzyme acting upon nucleic acid, okay, that is nucleases. Similarly the enzyme, special type of enzyme maltase, that is acted on maltose, sucrase that acted on sucrose. Okay, then urease that acted on urea. Okay, then lecithinase that we have already seen in the lipids. Okay, that acted on lecithin. Okay, tyrosinase. Okay, they acted on amino acid tyrosine. Okay, so that is why which substrate the enzyme is going to act, and simply a suffix ASC is added to name the enzyme. So this is the first naming and classification criteria. The next one is the type of reaction they catalyzed. Okay, which type of reaction the enzymes catalyzed. Okay, the enzymes are highly specific as to the reaction they catalyze. I told you that is one of the important difference between the chemical and biological. Although chemical specificity is there, biological catalyst, the enzymes are so specific in which substrate they are acting on. So this has necessitated their naming by adding the suffix ASC in the name of the enzyme. Okay, so which process, the reaction they catalyze, simply add the along with the reaction instead of the substrate, then they can call it as the enzyme. So hydrolase, they catalyze hydrolysis. Hydrolysis is a process, okay, the reaction, and the enzyme is known as hydrolysis. Okay, isomerization. Okay, isomerization is a process. The enzyme is isomerase. Okay, simply isomerase. ASC they have added. Oxidases, okay, oxidation, okay. Dehydrogenases, the dehydrogenation, okay, this is a process, okay. Transaminase, the enzyme is known as transaminase and the reaction is transamination. The enzyme, the reaction that carry out transamination is known as transaminases. Then transaldolation, okay. So the transaldolyses are the enzyme, okay. Then transketolation, okay. Transketolases are the enzyme. So phosphorylation, okay. Phosphorylases are the enzyme. These are common enzyme you have keep on. No, you are studying in your physiology or biochemistry or chemistry, microbiology, and all the metabolic reaction you might have heard all this reaction. Then the next one is the both the things combined. The first two things, okay, the substrate acted upon and the type of reaction they catalyze. So first two things they have combined. Here the both the substrate they are acted upon and the type of reaction what they catalyze consider into the criteria. Okay, so and based on that one they have given the naming. The enzyme succinic dehydrogenase, okay, that is an enzyme succinic dehydrogenase catalyzes the dehydrogenation. Here the dehydrogenation is the reaction and the substrate is succinic acid. So what it will do, it perform a dehydrogenation reaction where the substrate is succinic acid. Okay, so we are comparing the substrate utilized and the type of reaction that is involved actually. Similarly, L-glutamic dehydrogenase, okay, catalyzing the dehydrogenation reaction and that the substrate is L-glutamic acid. Okay, so that is why the substrate acted upon and the type of reaction. Similarly, many more examples you can see. This is the third classification criteria. The next one is the substance that is synthesized. Okay, so after the reaction, what is the name of the substrate? What is the end product what they have synthesized? Okay, so few enzymes has been named by adding the suffix ASC to the name of the substrate is produced or synthesized as a reaction of an enzymatic reaction. Okay, so example, rhodonase. Okay, so rhodonase is an enzyme that forms a rhodonate from hydrocyanic acid and sodium thiosulfate. Okay, sodium thiosulfates combines with the hydrocyanic acid, you will be getting a product called a rhodonate. Okay, so the name of the enzyme that is produced that is utilized in this step is rhodonase, because rhodonate is the rhodonic acid is the main important component. Similarly, fumarase that forms fumarate from a malate. Okay, malate converted into fumarate by an enzyme called a fumarase. Okay, so in which the substance that is being synthesized, what is the end products are synthesized due to this particular process. So based on that criteria also the naming of the enzyme can be given. Okay, then the chemical composition of the enzymes. Okay, so what are the chemical composition of the enzyme? So what the enzyme consists of? Okay, so enzyme, if the enzyme consists of only a protein part, okay, the protein part of the enzyme is known as apoenzyme. Okay, you have already discussed. Okay, and if the enzyme consisting of only a protein part, then such kind of an enzymes are classified into a particular group. Okay, example, pepsin, trypsin, urease, papain, amylase. Okay, all those things are the enzyme contains only proteins. Okay, then enzyme contains a protein and a cation. Okay, say for example a carbonic anhydrase, it contains Zn2 plus as a cations. Okay, or arginase Mn2 plus as the cation. Okay, tyrosinase copper 2 plus Cu2 plus as the cation. Okay, so the enzyme contains a protein and a non-protein part. The non-protein part is known as a prosthetic group. Okay, on the basis of the nature of the prosthetic group, in addition to a protein part, If the enzyme has a non-protein part, the prosthetic group is there, then based on that one, another criteria, another classification criteria is there. Okay, so on the basis of the nature of the prosthetic group involved, the enzymes are mainly 4 types. Okay, so one is iron porphyrin enzymes. Next one is flavoprotein enzymes. Next one is diphosphothioamine enzyme. Then next one is enzyme require other coenzymes. Okay, so these are the major types of classification based on the chemical composition. So iron porphyrin enzymes examples are catalyzers, cytochrome C, peroxidazes 1 and 2. Okay, here the protein part will be there and non-protein part will be there. Most of the case there will be iron. Okay, will be there, or any other cations will be present. Then flavoprotein enzyme, glycine oxidases, pyruvate oxidases, histamine, all those things are flavoprotein enzymes. Okay, diphosphothioamine enzyme, beta carboxylase, pyruvate mutase, all those things are comes under this particular group. Okay, enzymes requiring other coenzyme. Okay, example phosphorylase. Okay, and it requires an amino acid decarboxylase is required for that actually. So these are the chemical composition of the enzymes. Then the next one is the substance hydrolyzed and the group involved. So what is the substance in which it is hydrolyzed and what are the major groups are involved? Based on that one, there are 6 classification. 6 enzymes classifications are present. Okay, one is carbohydrate hydrolyzing enzymes. Okay, the carbohydrate hydrolyzing enzyme. Next one is protein hydrolyzing enzyme. Third one is lipid hydrolyzing enzyme. Fourth one is other ester hydrolyzing enzyme. Next one is oxidation reduction enzyme. Next one is miscellaneous enzyme. So like that, there are 6 group of classifications are present. So carbohydrate hydrolyzing enzyme, the enzymes are known as glycosidases. Okay, commonly known as glycosidases. We have already seen that enzymes in unit number 5. Okay, so we have already discussed about carbohydrate. Okay, so the glycosidases, the major enzymes are cellulases, amylases, sucrases, lactases, maltases, etc. All those things are they hydrolyze the sugar residues. Okay, in the 1,4 linkages. Okay, then beta glucuronidase. Okay, another group is beta glucuronidases. Okay, next one is the protein hydrolyzing enzymes. Okay, there are 2 types. If it is a peptide bonds, okay, if it is a peptide bond and non-peptide carbon nitrogen linkage. If it is peptide linkage, there are again 2 types, endopeptidases and exopeptidases. Endopeptidases, they cut it within the DNA. Okay, within the peptides. Okay, exo means outside the peptide unit. Okay, so that is in endopeptidases, that is present in the animals, and both animals and plants. Animals, pepsin, trypsin, renin. Okay, all those things, and in plants, papain, physin, bromelain, etc. Then exopeptidases are dipeptidases, tripeptidases, all the things are exopeptidases. Okay, then these are the based on the peptide bond. But what about non-peptide CN linkage? There are, that is known as amidases. Okay, example ureases, arginases, glutaminases. They are also a protein hydrolyzing enzyme, but the nature is not either endopeptidase or exopeptidase. They are non-protein CN linkage. Next one is the lipid hydrolyzing enzyme. They hydrolyze the lipids. Okay, so example is lipases, esterases, lecithinases, etc. The next one is other ester hydrolyzing enzymes. Okay, apart from this one, the ester hydrolyzing enzymes. Okay, phosphatase, cholinesterases, okay, chlorophyllases, then sulfatases, pectin esterases, methylases, etc. So these are the some of the important other ester hydrolyzing enzyme. Next one is oxidation reduction reaction, redox reaction enzyme, that is hydrases, mutases, oxidases, dehydrogenases, peroxidases are the oxidation reduction enzymes. Okay, next one is other enzyme, miscellaneous enzyme, that is a mixture, that is catalases, carboxylases, carbonic anhydrases, thiaminases, etc. Okay, these are the classification based on the substance, a hydrolyzed, and a group that is involved. Okay, then based on this is the very important thing. Last one, the overall chemical reaction taken into consideration. Overall chemical reaction, that is if they consider taken into consideration, then this is very and well studied classification criteria of enzyme. The chemical reaction catalyzed in the specific property which distinguish one enzyme from another enzymes. Okay, so this is the major thing. If the enzymes are classified based on the nature of reaction they catalyze, and they helps to differentiate the enzyme from one enzyme to another enzymes. Okay, and this is done in 1961 by International Union of Biochemistry, that is known as IUB. Okay, they used this criterion as the basis for the classification and naming of enzyme. Okay, IUB classification of enzyme, this is known as IUB classification. Okay, so they used it as this criterion as the basis for the classification and nomenclature of the enzymes. Okay, and the major features of this system of classifications are this reaction. The reaction and the enzyme catalyzing them are divided into 6 major classes, each with 4 to 13 subclasses. Okay, 6 major classes and each of them with 4 to 13 subclasses. Each enzyme name has 2 parts. The first part is the name of the substrate, and the second part is the name indicated, that is end with the ASE, and which indicate the type of reaction they catalyze. Then the next one is the additional information like the nature of the reaction if required, and it is required then it will be given in the parenthesis. For example, the enzyme malate dehydrogenase, which catalyzes L-malate plus NAD plus into pyruvate into carbon dioxide plus NADH plus H plus. Okay, this you know very well. And the enzyme designated as L-malate. Okay, so the enzyme designated as L-malate, that is L-malate NAD oxidoreductase, that is in bracket we are writing, you see the nature of the reaction, decarboxylation. Okay, so that is what L-malate then colon NAD oxidoreductase. Okay, and but this type of naming we are not using commonly. There we are using a trivial name, and these trivial names are very easy to understand. Okay, that is what I wanted to emphasize here. Then each enzyme has been allotted a systematic code of number called EC, or enzyme commission number. This is very important. Okay, so according to IUP classification, each an enzyme that represents a set of a number, okay, that is separated by dots, okay, and that is known as systematic code for a code number, that is known as enzyme commission number. Okay, generally EC number for each enzyme consists of a series of number which are 4 places. Okay, and what are the 4 places indicate? Okay, the first place is the number represent the major classes of the enzyme which belongs to. Okay, what is the major class of the enzyme? And the 2 median number which denote the subclass and their subclass of the enzyme within the major class. Within the major class there is a subclass. Within that particular subclass there is subclass. Okay, then the 4th digit represents the serial number of the enzyme within the subclass. What is the serial number of the enzyme within the subclass? Say for example, an enzyme EC 2.7.1.1. Okay, so what is the meaning of this? Then is EC enzyme commission number 2.7.1.1. So that represents the class 2, which is the transferase, and a class subclass. And I told you the first one is indicating the major class, that is major class 2 is a transferases. Okay, because there are 6 classes are there. In that second one is transferases. Under this transferase there is a subclass in 7. Okay, those enzymes which transfer phosphate group, that comes under subclass 7. And in that particular subclass there is one more subclass, that is 1, if that is a transferring phosphate and an alcohol group of the phosphate acceptor. Okay, they transfer the phosphate, that is why it is known as 7. And under that 7 subclass that phosphate acceptor is an alcohol group. Okay, and the last one indicate, the final one indicate the name of the enzyme. Okay, say for example hexokinase, okay, or ATPD hexo 6 phosphotransphorases. This hexokinase is a trivial name, and this systematic name is the ATP colon D hexo 6 phosphotranspherase. Okay, so that is clearly indicating which class everything it will come. That is what is the EC number, EC 2.7.1.1. 2 indicate what, 7 indicate what, 1 indicate what, 1 indicate what. 2 indicate the major class, which is a transferase. 7 indicate it transfer a phosphate group, Under that one, the first category is the phosphate group. It is the phosphate acceptor is an alcohol group. That is why it is first. Okay. Then the next one is the final digit of the enzyme. That is in that particular thing it is a hexokinase. So the number is hexokinase. Okay. So the first number indicates the hexokinase. So that is what I wanted to tell you. So these are the overall chemical classification taken into consideration. And based on all these things, as I said, these enzymes are classified into mainly 6 groups. The IUB classification of enzymes, there are mainly 6 types: oxidoreductase, transferases, hydrolyses, lyases or desmolases, isomerases, ligases or synthetases. Okay, so that is what these are. Okay, now IUB classification: oxidoreductase, transferases, hydrolyses, lyases, isomerases, ligases or synthetases. Okay, all those things. Okay, so name in itself it is there. What is this particular enzymes? Okay, so oxidoreductase are the that catalyze oxidation reduction reaction, the redox reactions. Transferases, the transfer. Okay, so they will transfer something. Hydrolyses, they will involve hydrolysis process. Okay, and desmolases, that is a cleaving process. Okay, lyases, isomerases, they classify, they perform isomerization. Okay, ligases, they will synthesize or synthetize, they will involve in the synthesis process. Okay, so that like that the name easily we can define. So one we will be discussing briefly. Okay, so here the enzymes earlier called oxidoreductase, I told you they gives oxidation reduction reaction called dehydrogenases or oxidases or peroxidases or hydroxylases or oxygenases. Okay, so these are the major enzyme which comes under, that is the earlier name of oxidoreductase. So what is this? This enzyme which brings out the oxidation reduction reaction between two substrates. Okay, between two substrates they brings oxidation reduction reaction. They catalyze electron transfer reaction. Okay, because when you are oxidation reduction reaction is happening, what is oxidation you know that, what is reduction you know that. They catalyze the electron transfer reaction. The enzymes catalyzing the oxidation reductions are the enzyme that catalyzing the oxidoreductions of CH-OH group, C double bond or ketone group, CH-CH group, CH-NH2 group and CH double bond NH group. Okay, so these are the major oxidoreduction reaction in which these enzymes involved. So in this particular enzyme there are mainly 4 subclasses. The enzyme acting on CH-OH group. Okay, CH-OH group of electron donor. Example, alcohol dehydrogenase. Okay, it's a trivial name. Second one is a enzyme acting on CH-CH group on the electron donor. Okay, if it is CH-OH, this one, the CH-CH group of electron donor, acyl-CoA dehydrogenase. Okay, enzyme example is of this particular subclass is acyl-CoA dehydrogenase. Next one is the enzyme acting on the heme group of the electron donor. Okay, the enzyme acting on the heme groups of electron donors. Okay, cytochrome C oxidase. Okay, so cytochrome oxidase. Next one is the enzyme acting on H2O2, hydrogen peroxide. Okay, as electron acceptor. Okay, then example is known as catalysis. Okay, so these are the subclasses, and what is the at least one example you should know. Second class is known as a transferase. This is the second class. So what is transferase? The enzyme which catalyzes the transfer of a group such as G other than hydrogen, okay, between a pair of substrate such as S and S dash. Okay, so they transfer a group, for example G other than hydrogen, they transfer between a pair of substrate S and S dash. Okay, the enzyme catalyzes the transfer of one carbon group or aldehyde or ketonic residues or acyl or glycosyl or alkyl or phosphorus or sulfur containing group. Either they can transfer carbon compound or aldehyde or ketone or acyl groups, okay, or glycosyl group or alkyl group, phosphorus or sulfur containing any group. They have 3 subclasses. One is known as acyl transferases. Example is choline acetyl transferases. Okay. Second one is glycosyl transferases. Okay. Example is a phosphorylase. Then third one is the enzyme catalyzing the transfer of phosphorus containing group. Okay, phosphorus containing group. Example is hexokinase. Okay, the first enzyme that is involved in glycolytic process. This is the second one. Third one is hydrolysis. Okay, the third one is hydrolysis. The enzyme catalyzes the hydrolysis of the substrate by adding the constituent of water across the bond in which they act on. Okay, so the enzyme which perform hydrolysis process, and by adding the constituents of water across the bond, they split. Okay, the substrate include the ester or glycosyl or ether or peptide or acid anhydride or carbon-carbon or halide and phosphorus nitrogen bond. So these are the major place where hydrolysis can happen. The subclasses are the enzyme acting on the ester bond, that is lipases. We have seen, isn't it, how triglycerides are broken by hydrolysis process by lipase. Then enzymes acting on glycosyl compound, example beta galactosidase. Okay, so that also we have seen. The enzyme acting on the peptide bond. Okay, the enzyme acting on peptide bond, pepsin, trypsin, thrombin and plasmin, all those things. Then the enzyme acting on a carbon nitrogen bond other than peptide bond. Okay, example arginase. Okay, so these are the some of the subclass and there are important examples. The fourth one is lyases or desmolyses. Okay, so they remove the groups. Okay, the enzyme which catalyzes the removal of group from the substrate by mechanism other than hydrolysis. Okay, and leaving the double bond. They leave the double bond actually. Okay, so that will cleave or remove the substrate by a mechanism other than hydrolysis, and they leave the group. Enzyme acted on carbon-carbon, carbon-oxygen, carbon-nitrogen and carbon-sulfur and carbon-halide bond. Okay, say for example this is CX, X is a halide and Y is another halide. Okay, C, X and Y. Here you can see the enzyme, they will cleave, they remove this one and this one. Once these two things, once X and Y is removed, okay, they have formed a group. Okay, they formed a component and this form a double bond. Okay, so they create a double bond. So that is the major important aspects of lyases or desmolyses. Okay, then the next one is their subclasses are carbon-carbon lyase, carbon-oxygen lyase, carbon-nitrogen lyases. This lyases subclass are carbon-carbon lyase, example aldolases. Okay, then carbon-oxygen lyase, example fumarases. Okay, and carbon-nitrogen lyase, example histidase. Okay, so these are the major types. So this is all about lyases. Next one is isomerases. So as the name indicates, this enzyme catalyzes the interconversion of optical or geometrical or positional isomers of various components, or the intramolecular rearrangement of atoms or groups in this isomeric form. You know that the protein, amino acid, carbohydrate, okay, and everything will be will be existing in the form of an optical activity, both a D variety or a L variety. So there is an interconversion of an optical, geometrical or positional isomerism by this enzyme, that is known as isomerases. Okay, there also have subclasses like racemases and epimerases, then cis-trans isomerases, then intramolecular oxidoreductases. Okay, so these are the major enzymes groups comes under subclass comes under isomerases, racemase and epimerases. We have seen that racemic variety. Okay, we have already discussed what are epimers and anomers. Okay, so here that will catalyze the process in which they catalyze racemization. We have discussed. Okay, the enzyme is known as racemase and epimerase. Example alanine racemase. Okay, an enzyme. Okay, that comes under this particular class. Okay, the next one cis-trans isomerase. So retinine isomerase. Okay, an example retinine isomerase. Intramolecular oxidoreductases, that is glucophosphoisomerase. Okay, glucophosphoisomerase. So these are the major isomeric subclasses of isomerase enzymes. Okay, then the last one is lyases or synthesis. Okay, synthetases. Okay, so as the name indicates, they synthesize. Okay, so enzyme catalyze the linking two compound by utilizing the energy made available due to the breakdown of a pyrophosphate bond in the ATP. Okay, they broken down the ATP and they utilize the energy and they perform the combining of two components. They synthesize, okay, certain reactions. So the enzymes, such kind of enzymes are known as ligases or synthetases. This is the last class. Okay, so enzymes catalyzing the reaction that forms carbon-oxygen bond, carbon-sulfur bond, carbon-nitrogen, and carbon-carbon bond, and this linking can happen between carbon nitrogen, carbon sulfur, carbon nitrogen, and carbon carbon bond. So there are three important classes here. The enzyme catalyzes the formation of carbon sulfur bond, example acetyl CoA synthetase. Okay, the enzyme catalyzing the formation of carbon sulfur bond. Similarly, enzyme catalyzing the formation of carbon nitrogen bond, example glutamine synthetase. Okay, you can see synthetase the name. Okay, so the next one, enzymes catalyzing the formation of carbon carbon bond, that is acetyl CoA carboxylase. Okay, so these are the common enzymes. All these enzymes we will be discussing, we will be seeing in the various biochemical pathways of the cellular process. Among all these 6 varieties, our till date, okay, as of the recent studies, over 2000 different enzymes are known. More than 2000 different enzymes they have identified, of which the major groups are oxidoreductases, transferases, and hydrolysases. Okay, these 3 are the major group. Others are also evident, they have a functional role, but they are not that much prominent actually. Okay, so these are the major type of enzymes. So these are the main 6 group of IUPAC classification: oxidoreductase, transferases, hydrolysases, lyases, isomerases, ligases, etc. Okay, so that's all, dear students. We can conclude today's class. Okay, so once again we can glance through the main components studied in this unit, in this particular topic. So we have already made an introduction about the classification of enzyme and what is the requirement of a classification, what are the classification criteria, such as the substrate acted upon by the enzymes. Okay, then the name, type of reaction what they catalyze. Okay, substrate acted upon and the type of reaction, both they have catalyzed. Okay, the substance that is being produced, okay, or synthesized. Then the chemical composition of the enzymes, okay, substance that is hydrolyzed and the group which is involved. Okay, the overall chemical reaction taken into consideration, the International Union of Biochemistry classification, such as oxidoreductases, transferases, hydrolysases, lyases or desmolases, isomerases, ligases or synthetases. Okay, these are the major important classification and nomenclature of enzymes. Okay, so mainly you can expect the IUB classification is very important. Okay, the major classification in the examinations. So these are the main textbook. You can refer Principles of Biochemistry by Leninger and Principles of Biochemistry, Fundamentals of Biochemistry by J.L. Jain. Okay, and all other textbook, there is any enzymatic biochemistry book, all those informations are commonly available. So thank you, dear students. Thank you very much for watching this particular class. If you are having any clarification, you can ask me. Have a great time.